make it very precise one and a accurate one. So today's speaker is Dr. Dinesh. I would like to introduce him. Dinesh is a board-certified laboratory animal veterinarian with strong research background in the field of cardiovascular and metabolic disorders. He completed his BBSc and AH program from the Bangalore Veterinary College and Masters in Veterinary Pharmacology from IVRA under the guidance of Dr. S.K. Misra sir. Then he moved to USA to pursue graduate studies in the field of cardiovascular pharmacology. During his PhD training in Dr. Nancy Ross Laboratory at University of Arkansas for the medical sciences and postdoctoral research training in Dr. John Scott's laboratory at the University of Washington, Seattle. He worked on to elucidate role of ion channel proteins in the pathophysiology of cardiovascular disorders. After postdoctoral research training, he joined the Department of Comparative Medicine at University of Washington, Seattle as a junior faculty and continued his collaborative research in the field of cardiovascular field along with completing his residency in laboratory animal medicine. In 2015, he joined the Amagen Biopharmaceutical Company as a research veterinarian and led principal animal model development and exploratory studies along with providing ultrasound-based imaging expertise to support in vivo studies. He joined the Pfizer in September 2017 and as a laboratory head at Pfizer Cambridge site, he has been providing animal model development support as well as non-invasive imaging support to Pfizer programs at Cambridge site. So with this expertise, he will be delivering our webinar. Dear friends, today we have right now 3 o'clock, but his early morning has got up. He is having 4 o'clock nearly, and he has taken that much pain for delivering a webinar for our benefit. So I think all of you will be benefited, particularly in whatever area he is going to speak, that is a preclinical imaging biomarker in drug discovery. So take maximum advantage of him, and whatever queries you have as narrated, you can put it on the chat box, and then he will be the questions will be picked up and he will answer. Still, if you have any query, his email ID will be provided and you can share, ask him any doubt you have, and he will be glad to satisfy you in a befitting manner. And thank you very much. Now I introduce Dr. Dinesh. Dr. Dinesh, please.
Thank you, sir. Can you see my slide?
Yeah, we can see you.
Okay.
We can see the slides also. Yeah.
Okay. Thank you very much for the ISVPT for inviting me to deliver a lecture on this topic. It's been my passion in terms of how we could use the advanced technologies to accelerate the drug development. So I'm going to share overall my goal is to try to convince at least a 2 take-home message for the audience, or the value in terms of the animal welfare, the value of preclinical imaging on animal welfare in terms of the 3R perspective, and also the value of imaging in accelerating the drug development, how one would use these type of tools at the early development of new drugs. So before I jump on to the actual topic, I would like to have a disclaimer. I'm as the president mentioned, I'm an employee of Pfizer, and all the animal procedures and also the data which I derived from the animal studies are thoroughly reviewed and approved from a Pfizer institutional Animal Care and Use Committee, and also some of the studies which are conducted at the CRO sites, they were all met the standards and the guidelines of our Pfizer. So I broke the presentation into a 5 buckets. So the first, I would like to tell the value proposition in terms of the preclinical imaging, what the value it brings specifically in a arm of drug development. And I will briefly touch on the most commonly used imaging tools, specifically on a translational imaging tools and their basic working principles. And I'm not going into so much detail, just to kind of high level how that works and what you are looking from, what we are getting out of from that the imaging modality. Because later in my presentation I have a couple of examples on those using those tools. So I thought it would be beneficial for you guys to kind of get to know the principles, and then I would kind of describe the value of the 3R perspective in terms of the preclinical imaging and the imaging in 3 pillars of drug discovery, and wrap up my talk with one of the best examples showcasing how that preclinical imaging kind of clearly translated into a clinical imaging part. So with that, I would like to give a unique example how information which is stored on an image in a spatial fashion, how one could kind of derive that information in a quantifiable fashion. So look at this image. It's it looks like a boat or a small ship parked or standing on a water in one of the a quieter harbor. But if you expand that image to a little further, you start noticing that oh, there is something not right. It's not as quite as what you would see from the first image. And if you expand further, you can clearly appreciate oh, there is a greater something is amiss at this harbor, and the people are trying to rescue. One of the sailor might have fallen into water. So further, when you look at expand that space from that imaging part, so you can see that oh, the person who is in the water is really in danger. So if you look at the whole image, you can clearly see even though he is in a danger, and some of his co-sailors are trying to help him out of the water and also trying to avoid the danger what he is facing right now. So why I'm trying to show this one, when you limit your view in an image, you kind of you're not going to get a full perspective of what's happening. And so once you broaden, having a better view of the image, you could clearly appreciate what might be happening in that space. So basically, you're trying to derive the information as a function from that space within that image. So this is one of the slide which I borrowed from my colleague who always try to convey that the information, importance of the imaging and what one would focus when you're looking at when you try to derive an information from an imaging. This is one of the painting from a great Bostonian painter called John Singleton Copley. It's currently displayed at Museum of Fine Arts in a greater Boston area. So similarly, you look at this image. It's a bulb, and there is something is probing at one end of the bulb, and you cannot appreciate much what may be going on before and after. So when you try to view how that bulb looks, few seconds or a little earlier than what the first image was showed, so you can clearly see a bulb is completely normal, and it's appears to be working normally. However, when you move forward from that first image, and you can start noticing is there is something is poking through the bulb, and the bulb is getting into burst. So what I'm trying to convey message is, in addition to that, deriving an information in a spatial, in a overall space of the image. So the time is also critical, because that gives you a lot more information in terms of the dynamic process that is happening in the body, or cellular level itself. So again, this is one of the you guys have seen this image a lot. It's kind of a death of a bullet, death by a bullet image. So with that, the death of a light bulb actually. So with what overall, so the information which is stored in a space of in an image in a spatial fashion, and also in a time, is a very valuable. With that, so depending, there are some certain criteria one needs to consider before selecting an imaging tool for their study. So as you guys might have heard, there are so many variety of a cool tool in the imaging space came in place and in utility. So they all come with a different sensitivity and a spatial resolution. So one has to make a trade-off in terms of what type of sensitivity you're looking, and also what type of spatial resolution you're trying to achieve in your imaging modality. So the good thing is, as the advancement happening in the all the technology and the digital space, and each of these imaging modalities are getting better in terms of getting a higher sensitivity and also better spatial resolution. So in addition to a sensitivity and also a spatial resolution of the imaging modality, so one need to think, what you're trying to look and what you're trying to derive as a readout from your animal studies. So whether are you trying to achieve anatomical information from an animal, or are you trying to look at a functional or a physiological level of information, or per se at the molecular level of information. So each imaging, this is a schematic again, some like a CT, which is a computed tomography and X-ray based imaging modality. That's exclusively more often used in trying to understand the anatomical information from an animal model or an animal, or some imaging modalities such as magnetic resonance imaging and ultrasounds. These are both, you can derive both anatomical and also the functional information. For example, the echocardiography, which is the ultrasound-based imaging, where you could get a different cardiac structure, how that functioning, and also how that size-wise also. So the molecular ones is such as positron emission tomography, and also some of the optical imaging. So when I say optical imaging, which involves the two most common modalities, such as HIVIS, you guys might have used. It's called in vivo imaging system, and the other one is called an FMT. It's a fluorescent molecular tomography imaging. So they both fall under the optical imaging bucket. So in a preclinical imaging, there are, as I mentioned, there are so many variety of tools available to utilize for the purpose of whether you're trying to derive an anatomical information or a functional information or even a molecular information. And again, you need to make a trade-off on the spatial and also on the sensitivity wise from each of these instruments. But one of the cool things, irrespective of what imaging you're trying to do, so they all bring into a 3R values to your study. So in terms of the 3R values, or what are the advantages is you can, if you try to use imaging as one of the experimental readout, which certainly helps to reduce the number of animals that one would use for your studies. And also, it's a non-invasive. What I mean by that, you don't have to, have any surgical or you don't have to go through any invasive procedure. Basically, you can anesthetize some of the imaging modalities and acquire those images, or even in an unanesthetized status too. And the beauty of this imaging modality are, longitudinal measurements. What I mean by that, you can measure a different time point on a study, so which would help you to kind of track whether it's a disease progression or an efficacy of a molecule, and a high throughput, and which really helps in terms of number of animals you could image. At the same time, the data can be analyzed and made available for researchers or as a scientist to kind of take a look and make a faster decision around the programs. So I know, that we have so many tools we use in a preclinical imaging, but in a drug industry, specifically when you try to choose an imaging modality that for a preclinical studies, we always, the highest priority is looking at whether is that imaging modality have any translational value, because this is kind of, the image you can clearly see. There are a lot more imaging modality accessible that can be used in an animal model, and including the optical imaging modality. But when you bring your focus to the, what are the common ones used in a clinic, and what are those imaging modalities that are approved in a clinical use? So one of the biggest chunk of imaging modality missing in a clinical space are an optical imaging. So there is a less value in terms of the optical imaging in the clinical perspective, in a clinical use. So the most commonly used clinical ones are certainly the computed tomography, PET, SPECT, MRI, and ultrasound-based imaging modality. So because of that, at least in a drug industry perspective, we try to use more a translatable imaging modality in a preclinical studies, so that you can, whatever the results you're seeing in a preclinical animal models can be relate to a clinic. So just again, as I mentioned at the beginning, I'm going to give you a brief overview on some of the most commonly used clinical imaging modalities and their working principles. And then, I will, some of the examples which I list later in the presentation involves using those imaging modalities. Because of that, just to keep everybody on the same page. So let's start with the X-ray. So most of you guys are familiar with the X-ray. Either just having you guys taken one yourself either for the broken bone or some other purpose. So what's the basic principle behind the X-ray imaging? It's a transmission type of imaging modality. What I mean by that is, when you look at this leftmost image on the slide, so you can see a puppet shadowed on the wall. You have a light source behind your hand, wherever is the hand position. So what basically, the light rays are blocked to transmit through that the hand. As a result of that, you're creating a shadow on the wall. So it's the same principle for the X-ray based imaging too. You have an X-ray source and also the subject, and as the X-ray passes through, because of the body tissue is composed of a different density tissues. Some tissues absorb X-rays and some scatter those. So those ultimately the image you get from the X-ray based imaging modality are based on the composition of that, are differences from an absorption and also contrast provided by this X-ray attenuation at the tissue level. So most commonly used these imaging modalities in a clinic for the purpose of mammography, or you guys might have heard fluoroscopy during the surgeries, or a DEXA, which is called a dual X-ray absorptiometry, which is used to measure the body composition. And of course, the most commonly one of the most commonly used clinical imaging modalities is a computed tomography as well too. So there is a slight difference in terms of the X-ray based, even though the computed tomography is a primarily a X-ray based imaging modality, there is a slight difference in compared to any other X-ray imaging modality. The difference in terms of, you could get a very high resolution, a 3D imaging of a subject by using this approach. So the basic principle is the same. Basically, you have a X-ray source, and also you have a receiver, detector basically, and it was placed in between the subject, and this X-ray source and also the detector are rotated around the subject on a different axis, so which generates thousands of images from that rotation during the image acquisition. And they have a cool algorithms which kind of stitches all the images together. At the end, you can construct a 3D image. So this is one of the examples from the study where we scan the rat, and you can clearly see, the different axes, and try to derive the anatomical information from those animals. I'm getting some annotation. Can I decline it?
Yeah, please do not honor that. Please decline it.
Okay, thank you. So basically, the computed tomography is a very robust tool which could give you a very high resolution 3D imaging, which could be used for defining some of the anatomical structure in the body. So one other imaging modality, the most commonly used in clinic, is an ultrasound imaging modality. So what are ultrasounds? In a core definition in the physical perspective, it's any sound frequencies that are higher than 20 kilohertz are considered as ultrasound. So I would like to bring your attention to the lower panel, like a schematic where I show that different frequencies of the sound waves, and within that sound waves, the 20 kilohertz or anything higher than that. So specifically, 2 to 200 megahertz are the most commonly, the most commonly used for the medical imaging purpose, whether it's a diagnostic ultrasound or a functional ultrasound. Sometimes there has been a clinical research of using a therapeutic ultrasound approach as well. It doesn't produce an image, but it could use to either treat or destroy a tissue using that the ultrasound imaging. So how does that work? How does that ultrasound creates an imaging? So when you look at this schematic, where you have a transducer which produces an ultrasound at a certain frequencies, and those ultrasounds travel through and hits any object, whether it's an animal or a non-live object. So it, after hitting that, it reverse, reflects back, and the differences in terms of the frequency and also the speed at which the speed of the waves, reflected waves, can be detected from this transducer probe. And based on that differences, the equipment has able to convert into an electrical signal and generate a 2D images based on that reflected waves. And again, in a very high level, when you use an higher frequency transducer, you could get a very good resolution image. But if you want a good depth of a penetration of an ultrasound waves, you would use a lower frequency ultrasound transducers. So among the ultrasound imaging modality, means there are wide variety of use. When you look at the ultrasound imaging, whether it's a ultrasound guided injection, or somebody who's trying to measure a organ size, or even detecting the pregnancy and monitoring the pregnancy, both in preclinical and also the clinical space. Ultrasounds are those are the imaging modalities used. But in addition to those, the most commonly we we use ultrasound in a drug discovery for the purpose of echocardiography. So again, echo means the reverberation of the sound waves, and this using an ultrasound in assessing cardiovascular diagnosis purpose came from Edler and Hertz in 1954, and since then there has been a lot of advancement in the transducers and also image processing. Currently, because of that advancement, currently the echo is considered as one of the most reliable and cost-effective imaging modality one could use to derive structural information on the heart, irrespective of whether it's in a right atria or a left atria, ventricles, or a right and left, and also in terms of the functional information from either in animals or also as well as in human clinical patients. So, in addition to that advancement I mentioned, there was one of the greater advancement happened in a recent decades were availability of the high frequency transducers. So, and also the high frame rate image acquisition that really enabled utility of this echocardiography or an ultrasound based imaging modalities in a preclinical studies specifically on rodents, because when you have a smaller animal size, as much as like a mouse, for example, it weighs about 25 grams, and you really need a very high frequency transducer to get very good images from images on those animals. So the next, I would like to give a brief overview on the positron emission tomography. Unlike X-ray, the PET is a emission-based imaging modality. So the basic principle behind is you have whether it's a large or a small molecules, which is labeled with a positron emitting radionuclide. So when it's in the body, it emits the positron. The emission of a positron releases the gamma rays in two opposite directions. So basically, in an actual setup, you have a scanner, a circular scanner around the subject. Upon injecting any radio-labeled molecule, you scan at the different time points. Basically, the gamma ray detector detects those signals and that composes the image. And this has been one of the most commonly used imaging biomarker in oncology space, and also in other fields as well. And also it's a very high value in terms of translatability from preclinical to the clinical imaging. And one of the advantage with this imaging modality is that it's a very highly sensitive compared to any other imaging modalities. So the other imaging modality, I would like to just give a brief. I'm not an expert on the magnetic resonance imaging, but I thought of, I have one image later in my presentation, thought of giving you a basic principle how that works is. So as you know, any subject or an animals or humans, even the body is composed heavily with the water content. So water has hydrogen molecules and their proton carrying molecules. And I would like to bring your attention to the figure B, where in a normal situation where you are not exposed to any magnetic field, these hydrogen molecules spin in their own frequencies and with the different axes. And what in an MRI imaging, what happens is, when you put a subject into a strong magnetic field, which brings all these proton molecules to a certain axis. So some are in the the vertical fashion, some are in the downward, depending on the axis of the magnetic field. So once you brings the uniformity of the axis of these protons by this magnetic field, it could send a radio frequency pulse, which basically flip that axis, because you are kind of sending an opposite energy. So and then after RF pulse, you stop an RF pulse basically, and you still have a continued magnetic field, and the change in the axis spun back to the B0 axis basically, and the time it takes to come back to the normal axis depends on the different tissue composition. So that information is used to kind of map and create an image basically from an MRI technology. So again, people spend a whole semester just studying on the magnetic resonance imaging. It's no more. I don't want you to consider this. This is just a brief introduction on how that magnet creates an imaging. So I thought of showing this schematic image for that purpose. So with that introduction, I would like to kind of bring back focus of my presentation to the value. So one of the first value, as I said, in the preclinical imaging, it brings the 3R's value. So 3R is reduction, refinement, and replaces. So this is in terms of the animal welfare perspective. As I mentioned earlier, the imaging modality enables us to acquire an image in a longitudinal fashion, which reduces the number of animals one would need for the study. And also, because it's a non-invasive approach, it falls under as a refined technology. And also, it's a translatable as well. And you can derive a wide variety of information from these preclinical studies, including functional and also anatomical, or even the morphological by a non-invasive approach. So basically, it has a very high value in terms of providing animal welfare when you use an imaging in a preclinical studies. So I would like to show one of the example how that imaging has really kind of brought a 3R value to an drug development industry. So to showcase, one of the example, I picked the program where we use imaging modality to support, which is like a fatty liver disease. You guys might have heard non-alcoholic fatty liver disease or a non-alcoholic steatohepatitis. This is a progressive disorder. I would like to bring your attention to this panel where you see, the healthy liver, and because of the high fat food intake or the lifestyle, the fat start accumulating in the liver, and you have a continued over accumulation of fat in the liver results into steatosis, and the persistence of this condition results into a fibrosis, and steatosis induced inflammation, and then it results into a fibrosis of the liver, and ultimately a condition called cirrhosis, or it leads to a cancer development. And the prevalence of this condition is very high in a Western population, as much as approximation, based on the rough approximation, over 300 million fall under the bucket of having a fatty liver condition, and about close to 100 million people with the liver fibrosis. And there are no drugs that are currently being approved to either treat NAFLD and NASH. So as a result of, there are a lot of the drug companies are working to bring a breakthrough medicine to these patients. So how this being diagnosed in a clinic? So the gold standard to diagnose this condition in clinic is a liver biopsy, and it's a very invasive procedure and also very unreliable procedure. So if you use the same approach in a preclinical model, and when you're trying to understand the target or running an efficacy study, we use a wide variety of diet-induced rodent models of NASH and NAFLD. So where, if we try to use an invasive approach such as a liver biopsy, which certainly increases the number of animals one need, for the study just because the biopsy itself is considered as a added variability may influence the disease development, and also um, mainly the increased number of animals that needed to run the statistically robust studies because of you're having a liver biopsy. So there was a real need for a non-invasive imaging approach to quickly assess the condition and use those the readouts to whether it's randomizing the animals at the beginning of the study or assessing the disease level and even the progression and also the efficacy of a compound during the development. So what we did was we try to utilize the ultrasound-based approach to use the basic principle of that ultrasound. As I mentioned in the beginning, you have the ultrasound waves, which it's the object, and the reflected waves are measured in the transducer, and then it produces the image. But the reflected waves, their frequency and the speed at which they come back, it depends on the density of the tissue. So different tissues reflect on the different speed, and that's kind of directly relates to echogenicity of the tissue, basically. So in a simple terminology, the higher echogenicity means in an image it looks a very lighter and brighter, and lower echogenicity means the image appears as a darker appearance. So, for example, the liver and the body generally is a low echogenic tissue. So we use the same principle to assess steatosis. It's the deposition of fat in the liver. So what happens is when the animal started putting on the fat on the liver, started storing the fat in the liver, which increases the echogenicity of the liver, but that can be seen as as a bright spot. And for example, below image, what you are seeing is a axial image of the longitudinal image from abdominal area where you have the kidney, and also this is the liver. So these both are considered as soft tissue, and you have a similar echogenicity at the normal condition. So once the animals start putting on a fat in their liver, the echogenicity gradually changes, but you don't see the change in the kidney. So basically, if you normalize the changes in echogenicity with respect to the kidney, you could determine the level of the level of fat accumulation in the liver. So basically, you are looking at the hepatorenal index. So for example, in the normal, you would see around 1 or less than 1 in terms of their hepatorenal index, and in case of disease condition, especially the fatty liver, that value is almost doubled. So this is one readout, one way you can measure the fatty liver by using an ultrasound imaging. And the other component of our spectrum of the NASH or NAFL disease is now the development of the fibrosis, or the accumulation of the fibrous tissue in the liver. So in addition to the measuring the hepatorenal index, there is a modality within the ultrasound technique called a shear wave elastography. So I would like to bring your attention to this, the schematic. The basic principle behind this shear wave elastography is, you have a transducer which sends a very high frequency ultrasound through the soft tissue, and this high frequency ultrasound, when it travels through the soft tissue, it reverberates the soft tissue and a perpendicular to that the axis of the high frequency ultrasound, which creates the shear waves basically within the soft tissue. So transducer have a ability to measure the speed at which these shear waves travels perpendicular to the high frequency ultrasound upon applying over the tissue. So basically, one could get a mapping of that shear wave and also the speed of the shear wave travel across the soft tissue can be used to calculate it's called a E modulus, so which kind of tells us how stiff the tissue is. So basically, if the tissue is stiffer, the shear wave frequency is going to be higher, and because it travels very fast in a stiffer tissue. So in a fibrous tissue, in a normal tissue, the shear wave velocity is low. However, once the liver tissues start accumulating the fibrous content or increasing the fibrous tissue, the proportionately the shear wave velocity is also changes. So basically, you're trying to look at indirectly the stiffness of the tissue, which is used as a surrogate marker for the fibrosis. And one of the cool thing about this, the imaging modality is, again, it's been recently approved in a clinic to use for a diagnosis of a human patient as well. So I would like to, just show you one of the example how we use this imaging approach to in a preclinical space to bring the value in terms of the 3R perspective. So we had a study with the animals under a control diet, and also the other group with the choline deficient, high fat diet with the choline deficiency, and also 1% cholesterol in their diet. So once you put them on this diet, and gradually we track the disease progression every 3 weeks up to 12 weeks. So these are the representative image, which I already explained. I'm not going again. However, when you look at this hepatorenal index values on the graph, the red ones from the deceased animal model, and the blue ones line graph from the control. As you see, there is a significantly increased hepatorenal index is seen with the CDHFT diet group, but you don't see the same type of changes with the normal animals. And we did correlate with the histological markers such as ILO red staining, and it correlates very well. So, again, the histology is considered some of the gold standard for measuring the fat content in the liver. So this gives us a very high confidence using this imaging modality to use in our preclinical studies. So again, we use the shear wave elastography to measure the fibrosis in these animal models. And I would like to bring your attention to compare the shear wave mapping between the control and the CDHFT animals. You can clearly see that, sorry, I missed having the heat map. However, you can see the clear difference in the coloring. So what you're seeing in a CDHFT dias with this yellowish blue coloring is due to an increased stiffness. So it is quantifiable again. You're measuring the E modulus, which is a stiffness of the tissue. When you look at this summarized graph, at 3 weeks, you don't see a much appreciable difference between the CDHFT diet and also the control diet. And the red ones are the diseased animal model, and the blue ones are the control animals. As they progress, you continuously see increase in their E modulus, the stiffness of the tissue, up to 12 weeks. And we did compare the shear wave values, the elasticity values, with the gold standard series red staining at the histological section. In the histological section, you can clearly see a very robust correlation between the percent PSR area positive for the PSR staining versus the elastography. So it really clearly showed the value of using a non-invasive imaging approach to assess the steatosis and also the fibrosis. After this validation, we have been using very extensively all our NASH and NAFLD studies, and we are that result really reduced the number of animals we needed for our studies, and also accelerated the decision-making process around on the programs, because you could get immediately the values, how the study is progressing. You don't have to wait for an histological analysis to complete, which generally takes a couple of weeks. So besides, the 3R perspective, the other, the value of the imaging is that, you can use for supporting the 3 pillars during the drug development. So as you guys mostly aware that, the 3 pillars of the drug development includes, trying to see exposure at the site, trying to measure the exposure at the site of action, binding to the pharmacological target, or expression of the pharmacology, like a pharmacodynamic readouts. So in a layman terminology, these can be defined as, does the drug get there? Does it stick to the target? Or does the drug do something at the target level? So I'm going to, next few slides, I'm going to give a one example of using a preclinical imaging to support these pillars. So the first one is, does the compound reach the target tissue? So we had a program called ALK inhibitor, lorlatinib. It's a second generation crizotinib. Crizotinib was approved in 2011 for the metastatic non-small cell lung cancer. Basically, this is a second generation compound, lorlatinib. What they are trying to achieve is, to see whether to increase the blood-brain barrier penetration of this drug. Basically, the purpose, overall goal was to, by increasing the blood-brain barrier penetration of this compound would bring that, therapeutic effect in the lung cancer patient who have a brain tumor. So these were the lorlatinib was labeled with a radionucleotide, and then used in a non-human primate studies where to assess how good that, the blood-brain barrier penetration of this drug. So what you are seeing is a panel A is a full body PET scanner from a non-human primate, and certainly you can clearly see immediately after the administration of the radio-labeled the compound accumulation in the liver and kidney, you can start seeing the signals. So, but when you look at the brain, because that was the anatomical region of interest, you can clearly see the signals are lighting up on the different parts of the body, and you can quantify that, how, in terms of the pharmacokinetic perspective, how fast it appears in the certain part of the brain region, and how fast it gets cleared from there again. So to take home message is certainly, this program has used this PET imaging to evaluate whether this, provides the information in terms of lorlatinib reaches the target of interest, which is the brain. The answer is yes. So the second one is, does the drug reaches the tissue or bind to the molecular target? So just I would like to give one more example to showcase, the value of the preclinical image in terms of the pillar 2 is, the monoglycerol lipase program, where, this is a serine hydrolase. As you guys may know, it's the key regulator of the endocannabinoid signaling, and the inhibition of this the enzyme has a multitude of benefits in terms of for the several neurological disorders. So what the team did was, they developed the MAGL specific PET ligand, and it helped to characterize the several MAG inhibitors using this PET imaging modality. What you're seeing is, the top panel, it's a MRI image of an animal primate, and the below are the PET images. Often, you get you don't get anatomical information from the PET. That's why, they're hiring the MRI to try to co-localize the signal with the anatomical location, PET signal with the anatomical location using the MRI. So the B shows that immediately after that administration of the MAGL specific PET ligand, that you start seeing a lighting up hot spots on the brain where that MAGL activity is very high. And then the below image currently shows that using a blocker which kind of completely blocks the binding of that MAGL specific ligand, PET ligand to the enzyme. So clearly, this clearly shows that the PET imaging gave a benefit in terms of understanding whether the drug reaches to the binding site or specifically to the molecular target of that MAGL. So the third pillar is that does it affect the downstream pharmacology? So for that purpose, I choose the one of the examples from our recent program where the NAFED program, where acetyl carboxylase inhibition may inhibit the development of the fatty liver and inflammation, eventually the fibrosis. That's the overall hypothesis. As you guys may know that acetyl carboxylase is a key enzyme regulating in the de novo lipogenesis process. So if you block that enzyme, would inhibit the development of the fatty liver and inflammation and the fibrosis. So we use the shear wave elastography imaging to quantify the liver stiffness as a surrogate markers of the fibrosis. And these are the representative images of the shear wave mapping from a control animal, a vehicle, and also the ACC high inhibitor, acetyl carboxylase inhibitor. You can clearly see that from a summarized graph showing that the differences in the shear wave elastography from 3 weeks before putting them on a diet which induces the disease, and the treatment group. And as you see in the gray ones, you don't see any appreciable change with these are the control animals. You don't see any appreciable change with the elasticity of the tissue. However, in the blue and also the red circled ones are the animals that are on a diet which induces the NASH and NAFLD, and the blue ones with the ACCI inhibitor, and which the treatment group certainly showed a significantly lower elasticity. And these readouts were well matched with the other histological markers such as alpha smooth muscle actin and an immunohistochemistry, and also the percent PSR staining. So this is one of the example using an imaging where it shows does our compound provide an efficacy, how it affects in terms of the pharmacology, and also the ultimate the experimental readout. So you can clearly see that the value of this type of imaging modality in supporting all three pillars. So I did mention that at the beginning, whenever we start thinking about using a preclinical imaging as a tool for deriving a valuable information from the animal study, we always think in a translational perspective on the priority is it going to be translatable. So I will next few slides I have one of the examples where the imaging modality being used at the preclinical stage, and then the same imaging modality has been used in the clinical research as well and in the clinic. So for that, I choose one of the example from the fluoridate thymidine labeled PET imaging, where it's been used in an oncology research as a biomarker. So the program I choose is palbociclib, which is a CDK4/6 inhibitor, and basically it inhibits the cell cycle proliferation. So what we are trying to use, it's FLT is not a new one. It's been extensively used, even both in a clinic and preclinical imaging to stage the tumor and also evaluate the prognosis and response to the therapy. So as I mentioned, when the cells are getting proliferated, so if you have a radio-labeled thymidine, it would be inserted into the DNA. Basically, once it's inserted, you can use the PET imaging modality to measure that signal from the fluorinated thymidine, which indirectly tells us, whether if the cells are really proliferating very high rate, you would see a higher signal because you're going to have a more fluorinated thymidine inserted into the DNA. So this is a preclinical study, one of the images I borrowed from the 2012 paper, where you can see these are the MDAM xenograft model tumors. These are the type of the mammary tumors. So the top panel, the HC staining from a vehicle-treated group, and the below on the CDK4/6 inhibitor from the Pfizer compound treated group. In a graph below, you can clearly see the IHC scores for each of these. These are kind of a proliferative markers. And upon treatment, 3 weeks post-treatment, upon treatment, you can clearly see, the gradual decrease in their signal. But, basically you started measuring after 2 days of the treatment, and then for about 72 hours, you can clearly see the sudden decrease in these signals on all of them at 6 hours and 24 hours, but it comes back when you stop the treatment to the normal level, and that means you continue to see the proliferation. So basically, this is an in vitro type of analysis. When you look at the PET imaging, you see the same trend. At the baseline treatment, you have the signal at the same level, and 24 hours, 48 and 72 hours, you see a PET signaling drastically dropping in a treated group with the CDK4 inhibitor, telling that basically you're having a low insertion of the fluorinated thymidine, and that means indirectly telling us the cellular proliferation is reduced. And in 72 hours, because you don't have any more treatment, so it's coming back to the normal, not the normal, higher proliferation stage, basically. So same imaging modality being used in a preclinical, clinical studies as well. So what you're seeing is on the top panel, the representative imaging from responding patients. So these number 1, 2, 3 are the tumor locations. These were kind of FLT PET again, and these are 3 weeks after the treatment, and you can clearly see those the signals are getting reduced in terms of their intensity. It's about 80% signal is already gone. So this clearly tells us that the effectiveness of this compound on inhibiting the tumor proliferation, and certainly it helped in a clinical study to identify the responders for this drug and the non-responders as well. So hopefully, so far with some of the examples, I was able to convince that the robustness and the value of this imaging modality in promoting the 3 Rs, specifically in terms of the reducing and refining and replacing the animals, and bringing in a value in an animal welfare perspective. And also, I hope I convinced that the imaging can be used to demonstrate 3 pillars of the drug development in a preclinical stages, and those can be translatable and brings a very higher value during the clinical studies as well. So this is a small list of acknowledgement, but there are a lot of people. Pfizer is a huge company, and there are a lot of people contributed on some of the studies which I listed in my presentation. I would like to thank everybody. With that, happy to take any questions.
Yeah, thank you so much, Dr. Dinesh. It was really a very good overview and very interesting case studies, and an eye opener for many of us who has very limited knowledge in this area. So we have few questions. Can we take it up now?
Yeah, sure.
Yeah. Okay. So the first question is actually some of the participants have already answered to the question, but I'll just again put it to you. Dr. Sumit Prakash Yadav has asked, can ultrasounds may be harmful for using in diagnostic use?
Can you repeat that question?
Yeah. Can ultrasound may be harmful for using in diagnostic use?
So far, I mean, again, it's depending on the frequency of ultrasound you use, but so far I have not come across any of such harmful effect of using an ultrasound as an imaging tool. So I would say no.
Okay. The next question is from Dr. Same person. Any limitations of ultrasonography in diagnosis in animal reproduction?
So it's been extensively used, even both in animal reproduction and also even in a clinic, because when somebody goes for a pregnancy diagnosis and monitoring their pregnancy, it's been extensively used, and I don't believe that it has such a harmful effect on diagnosing or monitoring the reproduction using an ultrasound imaging.
Okay. Then there are 3 questions asked by Dr. Anup Kumar. So I'll ask one by one. The first one is, what about cost? Is expertise required to run this system, this kind of imaging systems?
Yeah, that's a good question. Because we always think as a scientist we would like to have a coolest toys and try to get a nice images, and always the trade-off would be, I forgot to mention, besides spatial and also the sensitivity, one would also need to consider about the financial requirement or an infrastructure. For example, the positron emission tomography, PET, and the MRIs are expensive equipment and also expensive to maintain as well. And these are specialized techniques. They are not a simple one. You need somebody who has an expertise in image acquisition as well as the analysis, and sometimes you need some basic familiarity with understanding the function of the whole system itself, just to kind of troubleshoot. Among some of the things I listed, the MRIs are the most expensive and also most resource intensive in terms of the maintaining the equipment as well, because you need to keep those the magnetic source at a very low temperature using the liquid helium, and that itself requires additional infrastructure besides the equipment itself.
Okay. The next question is, can we quantify how much drug reaches to the particular target? Is it possible to quantify the amount of drug?
Yes, actually. So that's what I was showing on one of the slides where you have they quantified the signal. It may not be a completely accurate, not accurate in the sense it may not be as close one would see with the circulating levels, but at least it has a pretty good correlation. So based on that, you could extrapolate or estimate the amount of drug accumulation at the site of action.
Okay. If any surgical procedure required in animals, can we reuse the same animal specifically if it is pharmacokinetic study? Maybe for imaging techniques, he is asking whether any surgical procedure is required. If not, can we reuse the animal?
So there are no surgical procedure. So far, the imaging modality which I showed, we don't need any surgical procedures. Only thing I would think in is, if you are using any contrast agents, for example, contrast enhanced ultrasound or a contrast enhanced computed tomography, you need to place a IV catheter, intravenous catheter to administer those contrast agents to acquire an image. Other than that, there are no surgery required, and that's the beauty of this imaging modality, because of that, it's a non-invasive approach. You don't need to go through any invasive procedures to acquire those images. In terms of the reusing an animal from the pharmacokinetics, and it's a very broader question. Depending on there are a lot of things you need to consider. What compound are you measuring in a pharmacokinetics? And also how long it lasts and what's the impact it's going to have when you use a compound A, and then how long you need to kind of have those the rest period before you run the second one, so that the first the compound where you use the animals for the pharmacokinetics may not have an influence on the following kinetic studies. It's not a simple question. I think it requires somebody who has an expertise in the pharmacokinetics, and I'm not the one in that.
Okay. Then I think the last question which he has asked, you already answered, but anyway, still I will put it for. Can we use same system for preclinical and clinical studies?
So some you can use, but because of the rules and regulation around the standards you need to maintain, and at which you need to maintain these imaging modalities to use in a clinic, and the preclinicals are slightly vary. And I think I have seen people using MRI in a large animal species studies, where they try to utilize the hospital resources to run those studies in pigs. So other than that, and also maybe some pet imaging as well, because of the size requirement, you can use interchangeably between the animals and humans. But there is a lot of, you need to make sure the guidelines of the hospital let you use those imaging modalities for your preclinical studies as well. So
yeah, that's right. Yeah. So the next question by Dr. Pramod Bharani. He has asked, he said it's a very informative session, and many of them have also congratulated. His question is validation of these imaging techniques required before submitting to regulatory authorities? I mean,
yes.
Okay. So the next question is, does organ-specific biomarkers can play a role in finding out abnormalities?
Yes, actually. So, for example, to give an organ-specific biomarkers, so I can tell you, I can take an example, one of the example where I showed the MAGL activity, like serine hydrolase enzyme, where the PET labeled ligand were used. So we know that the MAGL is very active in a brain, and using that the PET labeled ligand and lighting up primarily in a brain, which kind of tells you that the anatomical location of that activity. So it is kind of specific to that region. Same way, there are so many ways you can creative in designing your studies to answer your questions in terms of the specificity, or if you're looking at an activity in a specific organs as well.
Okay, and one more question is how contrast agents and animal ethics are related. I mean, I'm also not sure about this question. Dr. Kautak Kumar Sardar has asked this question. How contrast agent and animal ethics? Maybe is it allowed ethical perspective in animals?
Yes, it is allowed. In terms of, for any of these procedures or any of the drugs you are trying to administer, whether it's a contrast agent or a non-contrast agent, it's always what the scientific values it brings in when you try to answer when you're looking through the ethical lens, So if as long as it brings the scientific value and not affecting on the the animal well-being, certainly it's allowed. But so far, for example, the contrast agents are so broad terminology and depending on the modalities. So, for example, some of the contrast agents which are used administer at the end before the necropsy to scan the tissues ex vivo. Okay, so those certainly not allowed during the actual in vivo part of the study, just because they may have a untoward effect on the animal well-being. That being said, some of the contrast agents, such as ultrasound-based contrast agents, these are very inert and they are kind of, air bubbles wrapped with a lipid layer, and you inject them and they stay in the circulation only in the vasculature. Air bubbles, since there's a higher echogenicity, you can measure the vascular perfusion or, vascular density, and the same contrast agents cleared within 20 seconds from the body through the expiration. So it's depending on what type of contrast agents one would use, and they have a different effect on the body. So those things are considered when somebody is evaluating use of this contrast agent during the animal studies.
Yeah. So I think that's all the questions, and I have one question from my side. It will be the last question. So, is there any role of these imaging techniques in toxicology, like drug-induced toxicities? Can we determine in advance? So we generally rely on the histopathology for the toxicity and as well as the serum parameters. Whether this imaging technique can be helpful in diagnosing or detecting the toxicities in advance?
So, yes. So I can give you two specific examples. So, for example, the echocardiography is an ultrasound-based imaging modality that's extensively used during the cardiovascular safety studies. When you have some scientific rationale, the target modulation may have an effect on the cardiovascular function or a structure. We use the cardiovascular, the echocardiography. It's not a by default type of imaging modality. We will not have echocardiography for every study, but only when required and when we have a real scientific rationale, those echocardiography imaging would be added to the cardiovascular safety studies. And the micro CT, not the micro CT, the computed tomography, the micro CT in a rodent, we call it. So that's been used for some of the DART studies, developmental and reproductive toxicology studies, to measure some of these organ or a bone formation, such type of things.
Okay, great. Thank you so much. So now I would request Professor Bhauser, Secretary of ICPT, to kindly deliver the vote of thanks.
Thank you, Dr. Srinivasan. At the outset, on behalf of the society, I'm highly thankful to Dr. Dinesh for making a very good, informative presentation on imaging modalities and its applications in the drug discovery with very good examples, which were very easy to understand, and it showed the importance of the imaging techniques in the drug discovery. Thank you, Dr. Dinesh. I'm also thankful to all the, especially the company where the Dinesh belongs, Pfizer, for permitting him to deliver a lecture on the imaging techniques. So once again, I'm also thankful to Pfizer for allowing him for the presentation. I'm thankful to all the senior participants, those who are present over here. Also, all the life members of the Indian Society of Veterinary Pharmacology and Toxicology, as well as all the other participants from the other life sciences, as well as from the industry and from the certain, trust like this Tox Gurukul also, Dr. Rao may be present. So there are a lot of senior people, they are present over here. So I'm thankful to all these participants who made this event a very successful. So we have around 150 participants at the peak. So we are thankful to each and everyone for remaining present during the webinar. And at last, I'm thankful to the president of the society for permitting us to conduct this webinar, as well as I'm thankful to the webinar team of Dr. Srinivasan, Dr. Ratnadeep, and others, those who have helped us to make this webinar series a success, and particularly this webinar a success. This is our first webinar with a speaker from the other than India, or say from outside the India. So we got it a first time experience, and we are thankful to Dinesh for making it happen at odd hours, and he has spared his time. So once again, I'm thankful to Dr. Dinesh for making this happen for the Indian Society of Veterinary Pharmacology and Toxicology. Thank you, everyone. Over to you, Dr. Srinivas.
Thank you, sir. Thank you so much for the kind words. And now I request the participants to kindly fill the feedback form. The link is provided in the chat box so that you would get the e-certificate. So I think that's all for today, and I may request President Dr. Thakur to give any final remarks, or should I end the session?
Yeah, yeah. It was very nice experience to listen Dr. Dinesh. Everybody has enjoyed and learned about the preclinical imaging biomarker in the drug discovery, as well as in the toxicology also, and very nicely explained about the queries those were put up by the participants. So as Dr. Bhavsar has already proposed the vote of thanks, I sincerely thank all, as well as Dr. Dinesh and Dr. Srinivasan and Dr. Ratnadeep. And if there is no more question, nothing to add, so I permit you to shut this webinar, close the webinar.
Okay, sir. Thank you so much. Thank you, Dr. Dinesh. Thank you, everyone.
Thank you.
Thank you.
Bye.
Nice having you. Yeah. Thank you. Bye.
Bye. Bye, Dinesh.